Stone. I'm the chair of the School of Natural and Social Sciences, and I'm so thrilled that you're all here today for our 5th year of doing a Darwin Day lecture, but actually our 4th lecture, because last year, unfortunately, we had a horrific snowstorm that prevented our speaker, Dr. Britt Koskella, from getting here from California. So this year we've luckily been able to bring her back, and we're so excited. We've waited a long time to hear this talk. So thanks so much for coming. Dr. Mark Jonas is here. Many of you know him because you're in class with him. He's going to introduce tonight's speaker, but I just want to alert you to our other 2 talks this semester. So the School of Natural and Social Sciences is pretty broad. We're the largest school at the college, and we have lots of interesting majors within the school. So our talks really span a wide range of interests. So I just want to alert you because we have 2 upcoming talks that are just going to be fabulous. One is on Tuesday, March 3rd, and it's called Doing Anthropology in D.C. An alum of ours, Jeanette Yarwood, who is a graduate of Purchase in Anthropology, is coming back to talk about her work as an advisor to the U.S. House of Representatives on Africa. So she is really sort of right heavily in the world of D.C. politics, but as an anthropologist, as her title says, and she's been doing that work for many, many years. So it should be a really exciting talk, and she's going to come back as a Purchase alum on top of it just being a fabulous talk to begin with. And then on March 24th, we're going to have a talk by Ben Smith, who is a former CIA operative, and he's going to give a talk on clandestine careers. So his talk is about, based in his experience in the CIA. He's no longer in the CIA. He has to get everything he speaks about here approved by the CIA still, so it's very exciting. So I really urge you to come to both talks. They should be fabulous. So without further ado, I want to introduce Dr. Mark Jonas, who's an associate professor of biology here and really entirely responsible for bringing the whole concept of Darwin Day to Purchase. It's an idea that isn't ours, it's not our creation, but it occurs at many institutions, higher education institutions, universities, and colleges. And Mark thought it would be a great idea for us to celebrate as well, and we've been doing that. He's been doing essentially all of the work around making that happen, so please give him your warm welcome.
Okay, thank you and welcome to Darwin Day 2020. It's not entirely true that this is my doing. There are many people who were involved in organizing the event, and in particular, Dr. Linda Bastone's unwavering support for outreach activities like this enables us to sit here and listen to a wonderful science talk on Darwin Day. So I'd like to first thank Dr. Bastone for supporting Darwin Day. Thanks also to colleagues from the biology department and people from the office, Amanda Zallo in particular, for helping to organize. And this event would not have been possible without generous funding from Con Edison. So a little bit of background on Darwin Day. D-Day, as it's called, is typically celebrated in early mid-February, because it coincides with Charles Darwin's birthday. He was born on February 12th, H.B. Darwin, yesterday, and he was born in 1809. And it's a celebration of Charles Darwin's contributions to society and, more broadly, achievement and progress in modern science. Darwin Day is commemorated by hundreds of academic institutions with community outreach events and is recognized by the UN, United Nations, and its members as a celebration of science and humanity. We're sitting in the Northeast here. We're sitting in the middle of one big Darwin Day festival. We're not the only ones doing this. University of Connecticut, Boston University, Harvard, Columbia, many of the CUNYs, as well as SUNYs, like Stony Brook University, have Darwin Day festivals. And it's rapidly spreading. Well, in the current so-called post-factual era, we need more events like this. I hope you agree. And you participating in Darwin Day, contributing to it by simply being here, sends a small but very significant message to the broader community that facts, evidence, and science still matter to us. Do you agree?
Yes.
I hope so. Okay. Those who didn't clap, you still agree, I think. Okay. I'll introduce our 2020 Darwin Day speaker. I'm honored to introduce Dr. Britt Koskella from the University of California, Berkeley. She obtained her PhD in 2008 from Indiana University, where she worked on co-evolution of New Zealand mud snails and their trematode parasites. She then received an NSF postdoctoral fellowship to work at Oxford University and University of California, Santa Cruz on the interaction between bacteriophages, bacterial pathogens, and plants. After her time in Santa Cruz, she returned to Oxford and then moved to the University of Exeter on a series of UK National Environment Research Council fellowships. During her time as a research fellow, she examined the role of phages, the role that phages play in shaping natural microbial communities living in the phyllosphere, above ground tissues of the horse chestnut tree, as well as the link between the tree microbiota and disease. Dr. Koskella is now Assistant Professor of Disease Ecology and Evolution at the University of California, Berkeley, where she combines molecular approaches and experimental coevolution to examine microbiome-mediated protection against disease, the importance of microbiome transmission among generations, and the role of bacteria phage coevolution in shaping the microbiome. Let's give a warm welcome to Dr. Britt Koskella.
All right, thank you very much for the introduction. I'm very excited to be here. I grew up just a bit north of here, so it's nice to see the silvery northeast sky again. I'm happy to report also that the CIA did not clear my talks. All my secrets are still in here. We get them. Okay. So I want to start, before I get to bacteriophages, I want to start by just discussing the amazing transformation in our knowledge that's happened in the last decade or so, which is we've known for a long time about microorganisms and bacteria, and we've been studying them for many decades. But the recent advent of sequencing technology has underscored that our world is a microbial world. We are just visitors in a sort of microbial soup. And that microbial soup is incredibly important to shaping everything that we thought we understood about the eukaryotic world. We're now appreciating the role that microbes play in nearly all of it. And so I'm starting with a couple of my favorite examples and the sort of extreme examples, one of which is the role that bacteria play in shaping our weather patterns, for example. So you might not know this, but when the snow falls or when hail forms, each individual snowflake or hail is forming around an ice-nucleating bacteria from the atmosphere. So bacteria do everything from actually shaping our weather to, of course, causing disease in the case of cholera or syphilis here. We've known about bacterial pathogens for a long time. And the change more recently has been the shift from thinking about bacteria as negative, antagonistic pathogens that we need to control and lysol our way out of, into thinking about them as a critical component of ourselves. So this idea that our microbiomes are important for our health, our reproductive success, and our general well-being, including our behavior. And this is just a nice example of the way we think about this now in terms of our whole selves, including these microbial cells in and on us. And the best estimate at the moment is we're about 50% microbe. So for every human cell on us or in us, there's approximately one bacterial cell. And as I'll get to in a little while, there's, for every one of those, approximately one viral particle as well, if not 10 times. We have much poorer estimates of viral diversity. And even things that, once the advent of this whole genome sequencing has happened, if we look at the human genome or any eukaryotic genome, it's not just that the bacteria live in and on us, they're also inside of our genomes. So our genomes are made up of DNA of bacterial, viral, and fungal origin. So this is just a nice example of that. So I want, now that we understand this sort of microbial soup, I'm going to take a step back and a little tribute to Darwin here. As Mark said, Darwin's birthday was yesterday, February 12th, which actually happened to be mine as well. I didn't find that out until after I was already an evolutionary biologist. So when I discovered that happy coincidence, it just reinforced that this is the field that I will always be in. It's the best biological. All right, so Darwin set himself a very difficult task. He had an idea that evolution could happen via natural selection. The simple idea that by small permutations, any organism, any change that allowed one individual to have more offspring than other individuals in that population would spread through natural selection. It was a very simple idea. And it's the type of idea that I love because once you learn about it, it's just so obvious. You know, why didn't anybody think about this before Darwin? Well, because it's only intuitive once you've thought about it, once it's been introduced. And this is true of many important scientific discoveries. But instead of, I feel like had that idea happened now, he would have written his book about microbes. Because actually, studying evolution in microbial populations is remarkably easy. And it's very easy, and hopefully I will now convince you, that evolution happens rapidly and constantly in microbial communities and populations. But he set himself, of course, a much more difficult task about explaining humans using the theory of evolution by natural selection. But let's put his ideas into the context of bacteria, where I think the same exact rules still apply, and it's a little bit, well, there's more concrete and specific evidence of exactly how and when it happens. And the most obvious example, which is very relevant to us all here, is the evolution of antibiotic resistance. And this is, again, it's this exact same principles that Darwin was thinking about. The simple idea is that if you start with a population of bacteria, a pathogen that's infecting us, for example, right? And you put it under selection, so now you take antibiotics to kill all of those cells, that any variation in that population, such that a few cells, one or a few cells, have resistance, just by chance, to that antibiotic, because evolution is chance. They have a mutation that happened to be there, for example, or they came in with a pre-adaptation. Those resistant cells will be selected for, so they will spread, right? They're the ones that survive after antibiotics. They then reproduce despite the presence of antibiotics, and then they become the new population. Okay? And this is, again, intuitive, it makes sense, and we can watch it happen. We watch it happen rapidly. I can do it in a test tube. I can prove that it happens within a day in a test tube, but it's happening all the time, including in bacterial pathogens of humans. And this is a really nice and important illustration of this. So what you're looking at here is the advent of new antibiotics, so the discovery of new antibiotics and when they first went into clinical use, and then how much longer it took before we observed resistant bacteria in human patients. So in other words, on average, about 8 years after antibiotics start being used in a clinical setting, resistance is found to be circulating in those populations. Now, luckily, it doesn't instantly mean that that antibiotic is no longer useful, because oftentimes these resistances happen within a patient but then don't have any onward transmission. And so the vast majority of pathogens circulating are still susceptible. But nonetheless, this shows you how quickly this evolution can happen, even at the population level. Even at the human population level, I should say. And we care about antibiotic resistance evolution because it has important consequences for human health. This is a bit outdated now, it's from 2014, but I think it really speaks to the point. This is just a map of the deaths that are attributable to antimicrobial resistant pathogens. So these are pathogens that we should be able to treat, but can no longer treat simply because resistance has evolved. So this is evolution by natural selection as Darwin had envisioned it. But what we now know is that evolution proceeds not simply by natural selection, but also by neutrality. There's neutral processes underlying evolution, but also that you can have changes that are non-genetic, like epistatic changes, changes in gene expression, maternal effects. These are all ways where a parent can impact its offspring in a way that's different than Darwin had originally envisioned, which is just heritability by genetic material. And so there's something called the modern synthesis, which happened in the 30s and 40s, I believe, has been revised multiple times since then. But the basic idea is that it took into account everything we had learned after Darwin about genetics and combined it with classic Darwinian evolution to have this modern way of thinking about evolution, the modern synthesis. And that's been very useful to us for some time, but that picture hasn't been totally complete. And there's many things about this that aren't completely inclusive, but the one I'm going to focus on for today's talk is this modern synthesis doesn't allow us to easily take into account the fact that microbes play a big role in our phenotype and that those microbes are actually transmissible. So in other words, when babies are born, especially when they're born via vaginal delivery, they acquire a lot of microbes from their mother. And those microbes are actually cultured over time through breast milk in the case of breastfeeding infants. There are oligosaccharides in breast milk that cannot be digested by the infant. We had no idea why they were there. And it turns out they're there because they feed the microbes. They change the microbiome composition of babies in a particular way. And so that didn't really beautifully fit in with this classic way of thinking about evolution. And so some researchers now are ascribing an even further revision here called the extended evolutionary synthesis. And one of the ideas behind the extended evolutionary synthesis is that biological inheritance is more than just genes. And that can include the microbiome. And so this has all been happening, and it's not, there are people, you might hear the term, if you get into this, the holobiont, and it's quite a divisive term. Some people really like it, some people don't like it as an idea, and that's the idea that the unit of selection in Darwinian evolution is not the individual organism, but it's us plus all of our microbes. And there's some utility to it, and some people would argue that there's less utility. But one way or another, we do absolutely have to take microbiomes into account if we're predicting how an organism will respond to change. So as you know, we're in a rapidly changing climate right now. Okay? And if we're going to predict how our crops or how natural populations are responding to climatic change, we have to take into account the microbes that they associate with. Because sometimes the speed of adaptation is actually a result of changes in the microbial community rather than changes in the organism, the population of the organism itself. And I think this is incredibly exciting because thinking about the world in this way unlocks, I think, unlimited potential in terms of how we can change it and solve problems. And so I'm just going to give you 3 examples, and then I'll get into the focus of today's talk, the phages. But the 3 microbiome examples I want to focus on hopefully will give you a sense of the breadth of what we can do now that we've unlocked this potential. So this first one, this is a nice paper highlighting the ways that microbiome research can impact sustainability in agriculture. And what you're looking at here is at least three different ways, approaches where you could manipulate or understand the plant microbiome. So you can breed for plants that have genes that optimize their own microbiome. So plants that associate with particular microbes that do what you want to do. You can also optimize the microbiomes themselves. So do essentially probiotics for plants, give them a microbial community that you think will have the function of interest. Or you can manipulate the sort of pesticides, microbial biopesticides, and think about everything that we do to plants in a microbial way rather than a plant-centric way. So those are the approaches. And if you start doing these, I won't go through this laundry list here, but you can see that there's a huge potential in terms of the application for that. And those include things like increased drought tolerance. There's very good evidence that we can increase drought tolerance of crops by changing the below-ground soil microbiota. All right. Increased productivity, increased disease resistance, and that's something that I study in my own lab, and we have very clear evidence that whether it be the seed microbiome, or a whole plant microbiome, that the presence of that microbial community is critical in stopping pathogen invasion and disease. Okay. And then on the right side, this is just basically showing, convincing you that this is absolutely in line with government priorities. All the things that we need to do to make a more sustainable future, there is a role for the microbiome there. So that's one example from the plant agricultural side of things. Coral bleaching from the oceans. This is just one example of ocean microbiomes and the importance here. This is a really nice empirical result showing that if you actually add these probiotic, these microbial communities, bacterial populations to coral in a lab setting, you can reduce coral bleaching. So this is just two examples. They have many more in the paper itself. But on the left side, you see at day 0, before they start the treatment for 2 different species, excuse me. And then they either don't treat them and bleaching occurs, or they do treat them with this probiotic and they can prevent coral bleaching. So I would say we're not actually there yet in understanding which probiotics we use, when we should apply them, how we should apply them, how we predict if they're going to be successful, whether in corals or humans or plants. But certainly this is just one piece of evidence in a very strongly building case, an evidence case, that this can work. And the third example, because we can't forget humans, is the human microbiome. So again, this is a beautiful review. If you're interested in this, I'll point you in the direction of this paper, where they're looking at the idea of if we can, and when we do, manipulate the human microbiome, what might we do? And they have, this is the epithelial cell of the gut. And so what you're looking at here is just, on the left, all of the ways that we know the immune system influences the microbiome. And by the way, the microbiome also critically influences the immune system, and that's on the right. And so the fact that the microbiome influences our immune system means that if we can engineer or tweak the microbiome, we can change our immune system. And that includes things like allowing the immune system to react when it needs to, so have pro-inflammatory responses, or in the case of a lot of autoimmune diseases, actually reduce gut inflammation by changing the microbiome. And so these little stars here are all the places in these known interactions in the epithelial lining where manipulating the microbiome might, by having an intervention, might change human health. Okay. So we live in a microbial world. This microbial world is itself evolving very rapidly, sometimes in response to things like antibiotics or other features, sometimes in response to our own diet as we change, we suddenly go vegetarian or suddenly start eating meat, for example. That's associated, has known associations with shifts in the gut microbiome. But if we want to untap this potential, there's a huge step we have to overcome, which is how we manipulate the microbiome. So much of what we know right now is correlational. So in other words, we can go out and we can compare patients that have colitis, for example, with patients that are not currently exhibiting colitis. We could sequence their microbiomes and we say, yes, they're different. But that doesn't tell us that it's a causative pattern, that the shift in the microbiome that we see is actually leading to colitis, for example. But now we're getting to the point where we can do some of these experiments, and I'll give you an example of this towards the end. But even when we can show a causative role, we have to be able to do it, to implement that causative effect. And that is a real challenge. And it's a real challenge for numerous reasons, but one of them is that there is no such thing as a healthy microbiome. All right? There is no single probiotic right now that everyone could or should take that's going to be healthy, correct for you. Everyone in this room has very, very different microbiomes. There are things that make them similar, but there are inherent differences, and sometimes that's a bad thing in the case of an underlying disease or syndrome, but most of the time it's perfectly fine. You still have a functioning microbiome. So all of that is to say that we're a long way from being able to precisely manipulate the microbiome because we don't know what we're going for. That's one problem. But the other side is we don't quite know how to do it either. So that is my segue into bacteriophages. So So let's think futuristically for a second. Let's think that we now know what the perfect microbiome for you is. Because I've just argued that it's not for everyone in the audience, right? But let's say that I could figure out, based on your blood type and your genetics and your environment and your diet, I've put all of those into my complex calculation, and I know what your microbiome should look like. How do I get you there? Well, I could give you antibiotics, but that's not going to work particularly well, because antibiotics have this broad killing efficacy. They would just wipe your flora completely, and we know that would not be beneficial. Bacteriophages, on the other hand, are very specific. So by that I mean that these viruses only infect particular cells that they can recognize and successfully infect. And this idea that phages might be able to be used to kill particular bacteria is not a new one. So first I want to make sure that we're all on the same page by what I even mean when I say bacteriophage virus. So what you're looking at here are 3 examples of life histories that a phage virus can take. But as a general statement, bacteriophages are viruses that infect bacterial cells. There are also viruses of archaea, there are viruses of fungi, and I'm talking here about bacteriophage specifically. There are 3 types of bacteriophages. There are those that bind to the host cell, so this is the outer membrane of a host cell. They inject their own DNA or RNA into the cell, and then they essentially hijack the reproductive machinery of that bacterial cell to stop producing bacteria and start producing viral particles. Okay? Once those viral particles are reproduced and assembled into new viral particles, they actually lyse the host cell. So they kill the host cell, and all of those bacteriophages are released into the environment in pursuit of new hosts. No active pursuit, viruses are not alive. Passive pursuit. If they happen to encounter a new host cell, they can lead to another successful infection. Some phages, however, do something that's a bit different. So what they do is, the first part is the same. They have to recognize a bacterial cell and inject their own DNA or RNA. But in this case, the DNA is actually incorporated into the bacterial genome. And this is what we call a prophage. And it can stay in that genome forever. And now it's just reproduced along with the bacterial cell as part of the bacterial genome. We care a lot about prophages because many important functions that we care about in bacteria are actually prophage genes. And that includes things like toxins, Vibrio cholera that causes cholera disease. The toxin that makes it so pathogenic is a phage. It's a phage-encoded toxin. Phages can also move around antibiotic resistance genes and all sorts of other important traits. And then there's these really neat but quite rare filamentous phages that act like a lytic phage, but they don't actually kill the cell. They just continually ooze out of the bacterial cell. For most of the rest of the talk today, I'm going to focus on what we call the lytic bacteriophages, those that must lyse and kill their host cells. So the idea that phages are important dates back to the 20s, and I'm going to come back to that in a moment. But we're realizing now that they're not just important in terms of bacterial killing in the environment or in terms of bacterial processes, but they might have actually been important in shaping our own immune system. And so this is work from Jeremy Barr, and he and others have put forward this idea called the bacteriophage adherence to mucus model of immunity. And it's just one example, but I think it's a really nice example of why we have to think about evolution in a more holistic way, thinking about the microbes as well as the host. And the idea is actually quite simple. So if you were to look inside your stomach at the moment, inside your gut, what you would find is that your epithelial lining is also coated with this mucus layer. And this mucus layer is incredibly important for stopping pathogens entering your blood. Okay. And so the depth of this mucus layer is important to our immunity, and it's really related to swelling and inflammation. Okay, so a normal gut, if you will, a non-inflamed gut, does not have much inflammation and also has this thick mucus layer. And this mucus layer we've known is important for many reasons, for blocking pathogen movement. But what they found is that there are more phages in this mucus layer than we would expect by chance. So it seems like one of the potential reasons for this mucus layer is essentially to trap bacteriophages, and that the bacteriophages form part of this defensive barrier, so that as pathogens or any bacteria try to penetrate this mucosal layer and enter the cells or the bloodstream, they can't because they have a high probability of interacting with a lytic phage and being killed. So that's just one example of why phages might be important in the human microbiome, for example. And I always use this as also an important opportunity to remind everyone in the audience that there's a lot we don't know about the microbiome, but one thing that we know is that more fiber is always good. And part of the reason is exactly because of this mucosal lining. So Justin Sonnenberg and others have been working on why fiber seems to be so positive for microbiome health. And it seems like one of the reasons is that there are certain classes of bacteria that eat mucus, and there's some that eat fiber. And those two are antagonistic. So if you feed your microbiome fiber, you have more fiber-degrading bacteria, and they outcompete the mucosal degrading bacteria. And so that leads to a healthy mucosal lining. Okay, so that's my plug to eat more fiber. Now, as I said, we've known about phages for a long time, and this is because our discovery of phages as bacterial killing entities predated our discovery of antibiotics. So in the early 1920s, Felix Durrell and his colleague Frederick Tort, they co-discovered these viruses that kill bacteria. They did not know they were viruses at the time, but they knew they were replicating organisms or entities that could kill bacteria. And so Felix Durrell and many others got very excited about this idea. Again, pre-antibiotic era, we had no way to control bacterial pathogens at this point in history, and he had just discovered something that clearly killed bacteria. And one of the places he studied these phages is in cholera patients. So what he would do is measure the stool, the feces of cholera patients, and in that feces you can culture cholera bacteria. So you can have some idea of how much bacteria they're shedding, so what the sort of internal pathogen density looks like. And he can also quantify, much as I still do in my lab now, how many viral particles there are that infect that particular bacteria. And what you're looking at here is a paper that he published in the Yale Journal of Biological Medicine, where he looked at 5 patients. Of course, now you would need far more replication than this to get published, but at the time this was quite groundbreaking. He studied 2 patients, here you see on the left, who died of cholera. And in the dark line, the solid line, you're looking at the amount of cholera bacteria that was being shed in their feces. And he compared that to how many phages there were that infected cholera in that patient. And he argues that in the 2 cases of death, right, either no phages ever appeared that infected cholera, or they appeared but didn't take off. And that allowed the bacterial population to take off and kill the host. But in 3 cases where patients recovered, what he saw was that the phage population either took off and seems to kill off the cholera bacteria, right, in both of these cases, or at least played a role in, or was correlated with decline of that pathogen. And again, this got people very excited, and it led to a number of trials where he would go to villages in India where there were cholera outbreaks and dump this phage cocktail, these phages, into the well. And sure enough, the epidemic, the local spread of cholera would stop in that village. And the only problem with this is that he wasn't thinking scientifically as we do today, and so he didn't have a comparison, a control. He didn't have any villages where he dumped an empty vial into the well. And the reason that's important is because if some strange white guy comes into your village and dumps something in your well, you might go to the neighboring well, which would also stop a cholera outbreak. So we still don't know which of those 2 mechanisms was driving this, but Nestle and others have invested a lot of money more recently in cholera trials for phages. So there's a renewed interest in this approach. Along those lines, here is a similar graph, but from cholera in Bangladesh. This is in the environment. Okay, so what you're looking at here is in the line, you're looking at the mean number of cholera patients, okay? And in the bars, you're looking at the prevalence of cholera phages that were observed in the local water samples in Bangladesh. And you might see a similar dynamic where the sort of crashes of the number of cholera patient cases are correlated with the presence of phage in the water. So again, this idea that phages might be regulating cholera outbreaks naturally without any intervention. And now the question becomes, how do we actually intervene and make this process more predictable with positive outcome? So this isn't just a correlational pattern. We can experimentally demonstrate this as well. So this is work from 2012 where phages that are active against Pseudomonas aeruginosa were used to, in a waxworm model, to see whether phage therapy for aeruginosa infections of cystic fibrosis patients could be successful. And what you're looking at here is how quickly, so the time to death of a waxworm that's infected with this bacterial pathogen, Pseudomonas aeruginosa. And Pseudomonas aeruginosa is notoriously hard to treat in cystic fibrosis patients, and it is often one of the leading causes of death in these patients. And what you find is that without any intervention, the worm dies quite quickly. But if you add either a cycle of bacteriophages, a mix, or just even a single bacteriophage that infects this pathogen, then time to death is much longer. So experimentally, you can show that the presence of phages can reduce disease and increase lifespan of the host. And this, of course, is a phage that's specific to that pathogen. Unfortunately, this doesn't always seem to work. So this is one of the first, it was just published last year, one of the first full clinical trials of a phage that is going on the market. So it's called Phagoburn, I'm not sure how to pronounce it, here it is. And this is a proprietary phage cocktail that is used to treat multi-drug resistant infections of burns. And what they did is they took burn wound victims and they either allowed them to go through standard care, which is typically antibiotics, or this phage cocktail. And the takeaway message is that the phage did work. So these patients, what you're looking at on the y-axis here is the reduction in bacterial burden. So how many of these bacteria, how the phage presence reduced bacterial growth of target pathogens. And phages do work. They do reduce bacterial burden, but they didn't work as well as antibiotics. So I'm just putting this in here because I think we have to be realist that phages are not a sort of silver bullet. They're not going to magically replace antibiotics if we do end up in a post-antibiotic era, which is possible due to evolutionary resistance. But they can certainly be part of the solution. And I will also just say that there are many individual cases of phage working miraculously to take patients off of their deathbed when they have sepsis and other things. The problem is that we don't have a clear understanding yet of how often that's going to be the case and what potential negative consequences there are. So if this is something that interests you, I will just say that there is a lot of room in this field for people who are interested in moving phage therapy forward. It is going to be part of our future of medicine. We don't know yet how large of a part it will play, but it's going to be there. And we need brilliant minds to help us figure out how to do it appropriately. The other nice thing about phages, though, is that it's not phages or antibiotics. And in fact, what seems like the best solution might be phages plus antibiotics. And this is work led by Paul Turner at Yale and many others who've been asking this question of actually, can we use phages to help us solve the problem of antibiotic resistance? And they had this idea that we know the mechanisms of antibiotic resistance. Maybe we could specifically use or even engineer phages that by selecting, through natural selection, by selecting on bacteria that are resistant to that phage, you're actually selecting for bacterial pathogens that are sensitive to antibiotics. So this is a classic evolutionary trade-off. You can't be good at everything. And in this case, there's a trade-off between being resistant to phage or resistant to antibiotics. And if you can't do both, then having both phage and antibiotics there could help us win this war against the pathogen locally. And here's two examples of how they show this can work. So what you're looking at here are two different antibiotics, Cipro on the top and tetracycline on the bottom here. and a panel of phages, of bacteriophages, again, Pseudomonas aeruginosa. And don't worry too much about the details. The important thing I want you to notice is that after they select on these bacterial populations for phage resistance, so after the bacteria evolve resistance to phage, they then looked at what that did to antibiotic sensitivity. And for everywhere you see a star, there was a significant increase in antibiotic sensitivity, which really suggests that there's a trade-off between resistance to antibiotics and resistance to phage. And so if you use, and in fact they've done this now in patients on multiple occasions, where patients that have very high loads of a bacterial pathogen that are drug resistant, you can treat them with phage and you see a resurgence of a sensitive bacterial population, and you can then hammer it with antibiotics once again that wouldn't have worked a week earlier. Does that make sense? And so I think that really is part of the future, as I've already described, of medicine. But of course, bacteria can evolve resistance to phages as well. And these are just 3 mechanisms by which bacteria evolve resistance to phage. The first of which is that bacteria might simply change their receptor. Whatever molecule was on the surface of the cell that phages were recognizing to start infection, if you alter that receptor, the phage can no longer recognize you. So that's a very clear path to resistance, and it's one we see in a test tube that happens within 8 hours in my lab. The other one is CRISPR-Cas resistance. So show of hands in the audience, how many people have heard about CRISPR-Cas genome engineering or editing? Okay, so this is a new technique where we can go in and actually edit genomes. We can change the expression of genes with this technology. But this is not a technology that humans invented. It's something that bacteria have been doing since the dawn of time to, in part, resist phage infection. It just took clever people like Jennifer Doudna to figure out that this would also be applicable to genome engineering or editing. And the third way here is through apoptosis. So some cells recognize that they're infected, and they essentially lyse themselves before the phage can reassemble. So they die, but they protect their neighboring bacterial cells from phage. So bacterial resistance can evolve through many of these mechanisms and many, many more. We're discovering more almost monthly, I would say. And I'm going to cover a couple of cases where we've observed this in the lab, in the field. I should say in the lab, we can observe this very quickly. I already mentioned this, but if we put a bacteria and a phage together in a test tube, that bacterial population will evolve resistance to the phage in less than 8 hours in some cases, certainly by a week. And when I first started my postdoc, I wanted to ask whether this happens in nature. And for anyone interested in these types of dynamics, this is one of my favorite. You can read about time shift experiments. They are the ultimate way, I think, to measure evolution. What you can do, and it's somewhat unique to microbes because you can't freeze a squirrel and resurrect it, you can certainly freeze it, is that with bacteria and with phages, we can collect them and put them in a freezer, freeze them in place, and then resurrect them at some point in the future. And because we can do that, we can do what are called time shift experiments. And the idea is pretty simple, and I'm going to illustrate it with a specific experiment. So these are the horse chestnut trees that Mark mentioned during the introduction. And what I did is I went to the park where I walked my dog, and I collected leaves from 8 different horse chestnut trees at the beginning of the season, and then every month through the growing season until the leaves dropped off at the end. And each time I collected them, I just put them in the freezer. And at the end of the season, I took all the leaves out of the freezer, I grew up all the bacteria, and then you can also, exactly what Felix Durrell did, you can take that leaf, smush it up, grow bacteria, and then filter with a very small 0.4 micron filter that allows phages or viruses to pass through, but not bacteria. And then you can do what we call a cross-inoculation. And it's a very simple question. The phages that are in that leaf, who can they infect? Can they infect this bacteria? And by infect, I mean lyse the cells. And this is visible to the naked eye. They form what we call plaques, which are just absence of bacterial growth. So I did this experiment. And the important thing to remember here is that I have bacteria from one time point, let's say July, and I cross it with phages from earlier in the season or later in the season from that same tree. And the reason you do all of this is because it allows you to, in a single experiment, monitor or visualize evolutionary change. So the bacteria from one time point, they have some level of resistance to the phage from that same time point. But my hypothesis going into this was that they should be more resistant to phages from the past. That if they've responded to phage-mediated selection, if they've evolved resistance, they should be resistant to phages that they've already seen in the past, right? And if the phages are able to counter-adapt to that resistance, so if what we call co-evolution occurs, then phages from the future should be more effective again. So whatever resistance was there in July shouldn't be effective against phages from August or September. I know time shift experiments are a bit of a mind bend sometimes, but does that generally make sense? Does the prediction make sense? All right, great. And this is what I found. So on average, in nature, exactly what we expect to happen based on what happens in the test tube seems to happen. Bacteria are indeed more resistant to phages from earlier in the season and less resistant on average to phages from later in the season. And I was very excited about this because it was one of the first pieces of evidence that phage-mediated selection leads to bacterial evolution in nature. All right. So that's what happens at a population level. And let me just do a quick time check, because I don't want to blast through. How much time do I have? I can speak fast through this last part or slow? Oh, perfect. Okay, great. So I can stay at a medium pace. That's fantastic. All right. So I've just shown you the impact that phages can have at a population level, right, which is what we think about with phage therapy targeting a specific pathogen. But now I'm going to zoom out, because I started this whole talk by saying that the whole microbiome is really important as a community. And so now I want to convince you that phages are important at that community level as well. They're not just important for isolated cases of pathogen invasion. They're important in regulating our microbiomes generally. All right? So this was work done by my graduate student at the time, Norma Morella. And what Norma did was something that had been done previously in seawater, but had never been done for a microbiome. But it's a pretty simple idea, which is you go out to the field. We work on tomato primarily in my lab as a model system, as a lab system. So she went out and collected tomato growing in an agricultural field at UC Davis. She washed off all of the microbial community that's living on the surface of that plant. So flowers, leaves, and stems. And then she pelleted all those microbes into a little microbiome pellet from that individual plant and separated that pellet into two things. So actually, I should say the pellet was mainly bacteria, so it's the supernatant that had the phage. So what you can do is, through filtration, separate that microbiome into viruses and bacteria. And what she did then, she ends up with two inoculations. One that's just what stayed up on the filter, that didn't filter through, that was the bacterial community. And we refer to it as phage depleted because all of the free viruses would have gone through the filter. And then we can also concentrate the phage viruses using this amplicon filter. And then we can have an inoculum that's just the phage. And we can then either spray plants with just the bacterial community, just the phage community, or recombine them. And importantly, when I say recombine them, I mean combine a microbial community back with the phages that were on that same plant at the same time. And when you do that, you can spray those onto new plants in a growth chamber, and you can ask a really simple question, which is how important are phages in shaping that microbiome as a whole? Okay? Does that make sense? So the first thing we did is we essentially counted bacterial density using a fancy machine called droplet digital PCR that allows you to tell with pretty nice resolution exactly how many bacterial cells are on that plant at the time we sampled. And what we were able to show is that a day after spraying tomatoes with either a tomato microbiome that was just bacteria or a tomato microbiome that was bacteria plus phage, that sure enough, in nature, in an agricultural field, there are phages there that kill the members of the community, okay? So here's the bacteria-only density, and that was significantly higher than the communities that grew in the presence of phage. So all of that is to say that in that field, at that moment we sampled, there are a lot of lytic phages, and those lytic phages are capable of killing the bacteria in the community. But you can do better than that, and you can sequence using 16S amplicon sequencing. So this is just a fancy way of culture-independent way of saying what bacteria are in that sample in terms of species, or what we refer to as operational taxonomic units, because species in bacteria are very difficult to assign. And what Norma did, in brief, is sequence this whole microbial community and asked a question. And the question is, did having phages there change microbial diversity? Why did we ask that particular question? Well, because on average, across all of the studies, at this point, tens of thousands, if not more, studies on the microbiome, one thing is resoundingly clear. On average, a more diverse microbial community is a good thing. So most of the time when you look at dysbiosis or disease-associated microbiomes, the signature is decreased diversity. Decreased diversity is bad in microbiomes, just like in our world. All right, so what we found was that when we put phages there, microbiome diversity was significantly higher. And that was after a week. So once we've allowed these microbiomes to really establish on the plant, the presence of phage led to higher microbiome diversity. And we had a reason to predict this, and I'm going to spend just a second on what that is. So phages, just like all parasites and pathogens, are expected to be under strong selective pressure to infect whatever type is most common in the population and in the community. If you're a pathogen that can infect the most common host, you can have more pathogen progeny, right? If you're a pathogen that can only infect one person in this room versus a pathogen that could infect 20, it's clear if you come back a month later which pathogen is going to be dominant, right? And that's what we call negative frequency dependent selection. So it's a very simple idea that by targeting common types, pathogens, and in this case phages, can maintain microbial diversity. They lead to a rare advantage. And that's exactly what we saw here. The other thing that's kind of neat is that having phages there decreased among host diversity. And so I'll tell you what I mean by that. So when you had phages present, the microbiomes of two different plants looked more similar than when phages were absent. Another way of saying that is if we sprayed on the exact same microbacterial community onto replicate plants, in the absence of phage, they all sort of went in the different directions. But when we had phages there, the phages sort of steered them all back together towards the same microbial community. And I think this is just one example of how we might use phages in the future to steer a microbiome in a direction that we care about, towards a healthy microbiome if we ever figure out what that is. And I'll end on this last piece of data here, which is not from myself, but it's a very recent paper, so I thought it would be interesting to this audience. This made the became Twitter famous yesterday or the day before. And it's a really nice study that shows, albeit correlationally primarily, that phages might be involved in stunted growth. Okay, so what they did is they looked at, they did collections, stool sample collections from children that exhibit classic stunted growth, and then children who did not exhibit stunted growth. And they sequenced all of the microbial communities, and then they sequenced all the viral communities. And I didn't show the data here, but much like, as I already said, in other diseases, stunted children tended to have lower microbiome diversity overall. That's a correlational pattern. It doesn't necessarily mean causation. It doesn't mean that those lower diversity leads to stunted growth, but it's a very clear correlational pattern. And what I am showing you here is that there was also an effect on the phage community. So if you look on the left here, this is just a pie chart of different types of phages. And you'll notice, I hope right away, that the stunted children group, whether they be younger, especially when they're younger, but even when they're older, have a different phage profile. So they have this one phage, the Cipitodviridae, that seem to dominate, whereas there was more diversity in different classes in the non-stunted children. Right. And this is just another way of visualizing that. Here's a nice stacked bar plot of all these different types of phages that we know about. that are important in the human gut. And hopefully you can see by comparing these that on average, whether there's a stunted child or a non-stunted child, they had different profiles of bacteriophages. And so again, I don't want to say that the phage diversity is causative, but they did, if you want to read this paper, they did do a nice actual experiment where they took these phages and bacteria and crossed them. But I'll leave that to anyone who's interested. But it's certainly to say that phages might be an important component of this story. Okay. So to wrap up, I hope that I've convinced you that the microbiome, if you weren't already convinced, that the microbiome is critical to host health and fitness, and that understanding microbiomes, whether they be soil microbiomes, atmospheric microbiomes, or our own microbiomes, is both important for understanding the biology, but also allows us to potentially do a lot in terms of preventing or changing the direction of biology or biological systems. And that phages might be important. So in terms of this unlimited potential, how do we start getting there? And I'm just putting in a couple of plugs here for things to keep your eye on, all right, if you're interested in these ideas. And one of them is fecal microbiome transplants. So has anyone in the audience heard of these fecal microbiome transplants? Great, okay. So this is a very simple idea that if you have a microbiome that's causing disease, that has a role in disease, that you should be able to wipe it out, usually with antibiotics, and replace it with someone else's microbiome, with a healthy microbiome. And these fecal microbiome transplants have not always, but when they work incredibly well. These are people that were on their deathbed and walk out of the hospital a week later after an FMT. And they've been, the amount of different diseases and disorders and syndromes that they've been shown to sort of cure or reverse is pretty astounding. The problem, of course, is that they don't always work. We don't know why they work when they do and don't when they don't, so there's a lot of work to be done. And occasionally, there are negative consequences. So if you just take someone, your neighbor or your best friend's microbiome and transplant it into you, you might inadvertently be transmitting some pathogens as well. And there's now some evidence that there's some pretty famous cases where that has been the case. So as a medical field, the do no harm principle, I think, is problematic if there might be a pathogen in there. So there's a lot of work to be done before we can use it, but nonetheless, this is certainly something to watch. And the same principles apply to whole ecosystems. This is a fantastic paper. It's fairly early, and this has been done in numerous ways since then. But what they've been able to show is that you can actually restore prairie land by transplanting soil microbiota from a healthy prairie land to essentially one that has been deserted, that is unhealthy, not functioning. So simply by transplanting soil microbiota and allowing those microbes to colonize below ground, that microbial colonization leads to prairie restoration. And again, this is just one example of many cases where we could maybe take a microbial approach to conservation as well as health. And certainly agriculture, as I began the talk with. And I really feel that phages are going to be important in this story. And we have barely scratched the surface of the potential here. But certainly we can use phages to steer and reshape both the native and resident microbiomes or a transplanted microbiome. And the beauty of phages that I think is different than the fecal microbiome transplant is that phages are not human pathogens. They will never be human pathogens. They cannot infect human cells. So you can transplant a phage community, and what it's going to do when it arrives is change the native bacteria. So in other words, one of the problems with probiotics is we don't know if that bacteria that you grew in the lab over there will even work in this new environment you're planning to put it in. It might not be well adapted to the human gut or to the plant root, right? But in this case, we're actually just taking the phages and changing the composition or densities of bacteria that are already there in situ and are well adapted. So I really do believe that there's unlimited potential here, and we have to do it with evolution in mind. And with that, I will thank you and take questions.